is Enzymes. This will be a guided C-note video, so handwrite your C-notes. As you go, there'll be key points in here, and just take notes on those key points. Once you're done, I want you to read your handwritten notes, clean them up a little bit, and type them into your notebook. There's already a pre-formatted page for you in the biomolecule section. It's under How Do Enzymes Work. Once they're typed up, take a screenshot and email me those results. Your goal for today should be that you understand what an enzyme is and why it's important in your life. So let's start off with this picture here. So you guys should be able to look at this picture and tell me what it is. There's some hints in here. It's got these spirals. It's folded over on itself. But what this is a protein. So remember, DNA makes RNA, and then RNA makes this protein. And this protein has a very three-dimensional shape. And hopefully remember that what's important about proteins is this shape, and that each protein has a different shape, and that shape determines what it does. So our first key point is going to be a review of translation. This is what you guys did last class, so remember what translation is. Translation is when I take RNA, or in this case messenger RNA, and I make a protein. So this was on that last video. Here's your ribosome, which is that protein factory in your cell. Here is the mRNA that was made by the DNA. So this would be one gene, right? It codes for one protein. And this mRNA is going to feed through this ribosome. And there's something called codons on here. So 3 bases. So here it looks like we have a G, an A, and a G. Well, that is one codon, and it's going to code for a specific amino acid. You can see here, these are my amino acids that have already been added. And you can see some other amino acids that are going to come in here and get added. And eventually this will become a functional protein. Hopefully you guys remember that there are 20 different types of these amino acids, and that the amino acids are coded for by that RNA that is made from the DNA. Right. Another key point here is going to be the structure of a protein. This is also a review. So there are 4 structures of a protein. There is the primary structure of a protein, secondary structure, tertiary structure, and quaternary structure. The primary structure of a protein is simply the order or the sequence of the amino acids in here. So every single protein is going to have a different order of these amino acids. So it might be serine, and then lysine, and methionine, and isoleucine, but another protein will have a different order, and that's because the DNA determines the order of these amino acids. So that's my primary structure. When I start bending or folding over that primary structure, or twirling it into this helix shape, that is the secondary structure. And then when I keep on doing that, so I fold it more, I twist it more, I turn into what's called a tertiary structure of a protein. Now this tertiary structure is a functional protein. It's got a very distinct shape, and it could do a job. Now some of these proteins are going to be added together with other proteins to form kind of like a super protein. That would be a quaternary structure. So you can see here's one protein, this kind of grayish one. Then there's a green one, which would be another protein, a black protein. But when they all come together, that would be a quaternary protein. So make sure you understand the different levels of the protein structure and focus on this one, that tertiary structure. This is that functional protein, because later on we'll show what happens when these proteins start to unravel, which is a bad thing. Right, our next key point is, what is an enzyme? So this is when we get into our topic for today. So an enzyme is a special type of protein, and what it does is it speeds up chemical reactions. So there's all these reactions that are going on inside of your body, and normally they would take a really, really long time to happen. But because we have these special proteins called enzymes, they could happen a lot quicker. And there's a word for this, catalyst. Catalyst kind of means to speed up. So enzymes are a catalyst. They speed up all of the reactions inside of your body, and they're basically responsible for all those reactions happening. Now there's two types of reactions that could happen. There's one called catabolism and one called anabolism. And this is all related to metabolism. If you remember what metabolism is, that's how we use and make energy in our body. Well, catabolism is when I break things down, whereas anabolism is when I build things up. And enzymes are going to be responsible for this. So you'll have some enzymes in your body that break things down, and you'll have some enzymes in your body that are going to add things together to make something bigger. So this would be like a polymer that I'm going to break down into my monomers, or this would be my monomers that I put together to make a polymer. So let's look at catabolism first. This is when I break things down. And probably the easiest way for you guys to learn about this would be to think about digestion. So we've already talked about this hydrolysis word. It's hydro, water, lysis means to split. So I'm using water to split things apart or to split these big molecules apart. And if you remember, starch here is a type of carbohydrate. It's a type of storage of this glucose. In fact, plants make this starch to store glucose, so they have glucose for later on. And we eat it, when we eat bread and we eat pasta, we eat this starch. And all it is a whole bunch of glucoses that are put together. And it's usually like thousands of them put together. It's really big. And as this goes through our digestive tract, our mouth, and into our stomach and our small intestines, we break this down. So there's an enzyme in here that's going to start breaking starch down, and it usually breaks it down into something called maltose. And then there's another enzyme later on that'll break that maltose down into this glucose. But starch here, that sugar, we have to break it down into smaller monomers, the glucose, so that we could actually get this glucose into our blood system. So remember, it has to get through the cell membranes in our intestines and then get into our blood. And then we're going to use this glucose later on to make energy, or ATP. Well, enzymes do that. Enzymes break this down. Without those enzymes, I wouldn't be able to break down this food, and it'd come in one hole and it would probably just go right out the other hole. So that would be a really good example of catabolism. And this is with anything we eat. So if I eat protein, I got to break it down into amino acids, or if I eat my lipids or fats, I got to break it down into the fatty acids so that I could actually absorb that stuff into my blood. So here's a different way to look at that same concept of catabolism. This purple here is my protein or my enzyme. And you can see that has this very specific shape. And something's going to fit in here. And in this case, this is like table sugar. It's called sucrose. So this would be like a glucose monomer, and this would be a fructose monomer. They're both a type of sugar, but this would be like that white table sugar. So when you eat it, there's going to be an enzyme that this sugar fits in, and it fits in perfectly, and nothing else is really going to fit in it. So one thing that's important about these enzymes is they only do one job. So the enzyme here, its one job is to break down sugar or table sugar. So this little sugar fits in this enzyme, and then it's going to split it apart into the two monomers. So a glucose comes out and that fructose comes out. Now these would be small enough to actually get through that cell membrane and absorb into our blood. And then our enzyme is free to do it again. Now the opposite of catabolism is going to be anabolism, or when I build things up. So remember, I use glucose to make energy. So I ate that starch, I broke it down into glucose, and then this glucose gets absorbed into my body, into my blood. And usually what I'm going to do is I'm going to send this to my cells, and I'm going to turn it, or I'm going to use it to make energy, ATP. Well, sometimes I get a little bit extra of this glucose, so I want to store it for later. And I'm going to store it in the form of glycogen. Hopefully you guys remember glycogen. Starch and glycogen are kind of the same thing. Starch is found in animal or plants, and glycogen is found in animals. So this is how we store glucose for later. So I took these glucoses, I used an enzyme, and I bonded them together. And that was that process of dehydration synthesis, which was a vocab word from the past. So here's our glycogen. It's just a bunch of glucoses bonded together. I'm going to store this in my liver or in my muscles so that I have access to glucose when I need it. All right. So what enzymes are is they are a special type of protein. And I already said this, but one enzyme will only do one job. And that's because the most important thing about an enzyme is the shape. So here I have the shape of this enzyme. So this blue thing is my enzyme. Well, it's going to do something with this little yellow part. So that little yellow part fits into that enzyme. And most likely, nothing else is going to be able to fit in there. So that enzyme will only work on whatever this little yellow thing is. So maybe I'm going to break it down into smaller parts, or maybe I'm going to build it up by adding two things together. Now, something that's kind of cool about enzymes is they're usually named for what they do. Like we talked about sucrose, which is that table sugar. Well, the enzyme that breaks it down is called sucrase. Same with protein. There's an enzyme that breaks down proteins that we eat, and it's called protease. So protein, protease. Lipids, there's an enzyme called lipase, so lip. DNA, how I replicate it, I use this thing called DNA polymerase. And poly should make sense to you. It means a whole bunch, right? So one of the other things that's kind of cool about enzymes is most of them, not all of them, but most of them end in the ASE. So sucrase, protease, lipase, DNA polymerase. So if you guys are ever reading some really big, weird, crazy science word and it has ACE on it, well, you should automatically assume that it's going to be an enzyme. So a type of protein that's going to speed up a reaction. So let's look at an enzyme. So this is what an enzyme is. It's got a very specific shape. It's a type of protein, that tertiary structure of that protein. And then something's going to fit in here. And these are vocab terms that you're going to need to understand. So whatever fits into this enzyme is called a substrate. So if the substrate is glucose and I'm going to build it up into glycogen, well, that glucose would fit in here and probably nothing else would. So our substrate fits into our enzyme, and where they fit together, that is called our active site. So whatever's going to happen, if I'm going to break it down or if I'm going to build it up, it's going to happen right here in this active site. All right, so once again, our substrate is anything that fits into that enzyme. It doesn't matter if I'm breaking it down or building it up. Whatever fits in that enzyme is called the substrate. And it's anything, starch is a substrate, protein, glucose, lipids, amino acids, fatty acids, whatever fits into that enzyme is going to be that substrate. Now, enzymes only fit one substrate. So we like to kind of say they're like a lock and a key. So the enzyme would kind of be like our lock, and there's only going to be one key that'll open it, right? So a key has to have that very specific shape, and it'll fit into that lock. So our key is like a substrate, and the lock is more like that enzyme. So they like to say it's like a lock and key. All right. One other important thing about enzymes is they are never, or I won't say never, but they're not used up. So we've seen this picture before, but I have my enzyme and I took this sugar and I broke it down into its monomers. Well, that enzyme is now free to do its job again. So it's just going to keep doing this cycle. So it is always there looking for the substrate. So here's that enzyme right here, and it's got a very specific shape. Well, something fits in it, and In this case, it's another sugar. It fits in it. I'm going to split it apart. I'm going to break it apart and release it. And then that enzyme is going to keep doing it. So they're not used up, and that's kind of important. Our next key point is just to review that vocabulary. So write down enzyme vocabulary and make sure you record the definitions of these 4 things. The enzyme is that helper protein that speeds up reactions. That substrate is whatever fits into that enzyme. The product, which I haven't mentioned yet, the product is what comes out. So whatever goes in is the substrate. Whatever comes out is the product, and that should make sense. Product is produced. And then where they come together, the substrate and the enzyme, that's called your active site. All right, so the next key point is something called activation energy. And this is probably the most complicated thing for you guys. What activation energy is it's the amount of energy I need for a reaction to actually happen. And you're probably going to see some sort of graph like this on your test. So here is the beginning of my reaction, and then this is whatever comes out, the product. Well, for this reaction to happen, to turn this reactant into a product, I need a little bit of energy, and that is called your activation energy. What an enzyme does, an enzyme lowers this activation energy. So with an enzyme, I wouldn't need as much energy for this reaction to happen. And I like to use like a ball. Like if my cursor is a ball, well, it takes a lot of energy to get up to the top of this. But once I start going down, I don't really need any energy anymore. So the energy it takes to start this reaction is called activation energy. And here it is with an enzyme and without an enzyme. So this blue line is going to be my reaction without the enzyme, and then this red line will be my reaction with the enzyme. So I start the same place, same reactant, so maybe glucose, and then the same product. Maybe I'm making that glycogen. Well, if I don't have the enzyme, I got to put in this much energy. That's quite a bit of energy until that reaction happens. Well, with an enzyme, I only need this much energy. So it's about half the amount of energy, right? This much energy compared to that much energy. So this reaction is going to happen a lot faster because it doesn't need as much energy to get up here. And you could use that ball again. It takes a lot of energy to push a ball up to the top of this hill. Not as much energy to push the ball up to this hill. So this one's going to go faster. So that's how enzymes speed up the reactions in your body because it decreases activation energy. All right, so our next key point is denaturing. And what denaturing is when I unravel my enzyme, the protein. So nature, that's a good thing, right? Well, if I add D in front of that, that's kind of a bad thing. So I'm going to make it unnatural. It's not going to work right anymore. And there's a couple of things that will affect how well your enzymes work. And the number one is kind of common sense. If I have more enzymes, obviously the rate of the enzyme action is going to be a little bit higher because it's easier for those enzymes to find the substrates. Likewise, if I have more substrates, it's easier to find the enzymes, so my reaction rate is a lot higher. Now, the things that are going to cause denaturing or unraveling of my enzyme is going to be temperature and pH. And what happens is if my temperature gets too hot, then the shape of this enzyme is going to change. So here's my enzyme, right? So I have all these amino acids put together in a certain shape. It's going to do some sort of job. Well, if it gets too hot, the shape will change, and then whatever's supposed to fit in there, the substrate, won't fit in there anymore, so it can't do its job. And the same thing will happen with this, pH. Now we've mentioned pH before, but it was a long time ago. What pH is, it's kind of that acid and base balance. And there's a scale for it. So a pH of 0 all the way up to a pH of 14. Well, right in the middle is called neutral. So at 7, it's neutral. And if it's anything less than 7, that's an acid. Anything more than 7, that's a base. And the closer I get to the edges, so the closer I get to 0 or the closer I get to 14, the stronger they are. And you can see some examples here, like water is pretty neutral. Lemon juice is pretty good acid, but then hydrochloric acid, or the acid in your stomach, that's pretty strong acid. Same here, like milk of magnesia, it's got a base, but then if I go down here to lye, that's a really strong base, and if I drank a whole bunch of hydrochloric acid, I'd probably kill myself. Same thing here. If I drank a whole bunch of lye, I'd probably kill myself. Your body is right about in here, about 7.35 or so. And if it gets much lower than that, that's really bad. If it gets much higher than that, it's really bad. And that's because your proteins are going to denature. They're going to unravel. So let's look at protein a little bit more closely. I mean, temperature more closely. There is an optimum temperature for your enzymes and your proteins. And in the human body, that's the normal body temperature, like 97, 98.7, something like that. And as long as it stays around there, 97 to 98.7 or so, it's good. Yeah, that's where your enzymes like to be, and that's where they're going to work best. Well, if I raise your temperature, like in humans, if we get a bad fever, like 103 degrees or higher, what happens is your proteins or your enzymes can start to denature. That means they could unfold and lose their shape. Well, if they're the wrong shape, they can't do their job anymore. Now, if I lower the temperature, they don't denature. It stays the same shape, but my molecules are going to move a lot slower. And if they're going slower, that means my reaction rate's going to decrease just because there's fewer collisions, or the enzymes can't find the substrates as easily. So looking at this little graph here, so my reaction rate is highest right here at the temperature of 37 degrees. And that's what a body temperature should be, 37 degrees Celsius. Now, we always change it to Fahrenheit. That's why we say 98.7. But 37 degrees around here, Celsius, that's where our body temperature should be. If my body temperature goes up, well, the reaction rate starts going down. And the reason it's going down is because some of my enzymes are unraveling, they're denaturing. And that's a bad thing. Well, here, if my temperature goes down, my reaction rate also goes down, not because they're denaturing, just because I have less collisions, or it's harder for those enzymes to find the substrates. All right, let's look at pH a little bit more closely. So any change in pH will cause my protein to denature. So, our body is, like I said, about 7.35. If our pH drops much below 6, well, all these proteins inside our body will unravel and stop working, and you could die from that. Same thing if it gets too high. If my pH gets like 8.3, well, you're likely going to have some very negative health effects, including up to death. Now, there are parts of our body that have a really low pH, which means it's a strong acid, like our stomach. Our stomach acid is very strong, so the pH is really low. Well, there's an enzyme in your stomach that pepsin, works at a really low pH. But if I put this pepsin in a normal pH, that pepsin will stop working. And here, like trypsin, it's in your intestines. Normally, it needs that pH around 8. Well, if it gets into your stomach, well, that trypsin's not going to work. It's going to denature, and it won't be able to do its job. So the most important thing about enzymes to remember is the shape is what determines what it does. So each enzyme has a very specific shape. It does one specific job. If I change that shape, then it can't do its job anymore. All right, here's some examples of some enzymes. You guys don't really need to write this down, but here's our substrate, like lactose. Maybe you guys have heard of this, and we've talked about it in class, but this is like milk sugar, and maybe you've heard some people are lactose intolerant. And that means that they don't produce this enzyme, lactase. So when they drink this milk sugar, they don't break it down, and then it gives them some negative effects. They get nauseous or they have diarrhea or something like that. Um, Here's maltose, which I mentioned earlier. There's an enzyme called maltase that breaks it down. And all maltose is two glucoses put together. So that maltase will break it down into single glucoses. Cellulose, hopefully you remember what that is. That's one of my carbohydrates. This is what makes up cell walls in plants and bacteria. Um, well, cellulase will break it down. Humans, we don't have this enzyme. We don't have cellulase. So sometimes when we eat some vegetables, we don't digest it, right? It goes through us. That's called fiber. Well, like a cow has this enzyme cellulase. So they're able to break down cellulose and get the glucose out of it. Whereas us, it just kind of goes right through our system. Lipase, that breaks down lipids. Amylase, that's a protein that breaks down starch. And there's amylase in your mouth. So when you start eating sugar in your mouth starts breaking down that sugar. Then it'll finish later on in your intestines with this maltase. Protease, that breaks down proteins. It's going to take a protein and break it down into amino acids. Remember the 20 types of amino acids. In fact, I wish that was written right here, amino acids. All right, so here's a summary of everything. Most important things, enzymes are a special type of protein, and all they do is speed up reactions. They have one job. So one enzyme will only do one thing, and there's one gene that will code for that enzyme. The shape of the enzyme is what's important. If that shape changes, the enzyme won't do its job anymore. The substrate is what fits into the enzyme at that active site. Whatever comes out is called the product, so it's what is produced by the enzyme. Activation energy, and make sure you guys understand this. Activation energy is the energy I need to start a reaction, and that my enzymes decrease activation energy, or how much I need for that reaction to happen. High temperatures can denature your enzyme. It can make it unravel and change shapes. So it goes from that tertiary structure back down into like a secondary structure or even a primary structure. The wrong pH will also denature that enzyme. And then enzymes usually end in ACE. Now that's not every one of them. Some of them don't, but most of them end in ACE. And they're usually named after what they work with. All right, so that is the summary or everything you need to know for the basics of enzymes. Enzymes speed up reactions. They're a type of protein. Shape is what is important. Make sure you read through your notes, clean them up, make sure they make sense to you, and then type them into your notebook. Screenshot your typed up notes and email it to me. Thanks.